Thank you. Is this good? Closeness to the mic and, yeah, okay, great. Right, okay, so thanks so much to the organizers for giving me the opportunity to talk about our work today. So over the past few years, me and my colleagues at Lawrence Livermore have been using single-cell RNA sequencing coupled with animal models of infection, mouse models of infection, to build an atlas of immune responses to diverse pathogens. And we've been mostly using viral pathogens, although one notable fungal pathogen that I'm here to discuss today. And by gathering this data, we've been starting to do comparative studies looking at responses to different pathogens in order to identify shared and unique responses to the different pathogens. And we think that single-cell RNA sequencing is an ideal tool for doing this kind of study, as you can probe the transcriptome in an unbiased manner on a single cell level. So you don't need to know what you're looking for in order to be able to measure it. So our overarching goals with this amassing of this data are, broadly speaking, to understand mechanisms of host immunity that could be manipulated to improve infection outcomes. Specifically, as it relates to coccidioides infection, we're interested in better defining the immune vulnerabilities that underscore severe infection, as well as identifying potential blood-based biomarkers of fungal infection that could distinguish it from other types of respiratory infections. And so I know we saw some single-cell data in the previous talk, but I wanted to take a minute just to orient people to how we look at and visualize single-cell sequencing data. So each dot on these plots represents a single cell. And in these UMAP plots on the left, you see the cells are color-coded by cell type. And we can also color-code the clusters by sample type. So when multiple samples are merged onto one plot, you can color-code them by the sample that cell came from. We can use this type of analysis to identify distinct cell populations as well as subpopulations within broader categories of cells, and specifically using subclustering. So you can look at, if you take that broader category of just macrophages and monocytes in this example, and you isolate those and subcluster them, you can get higher resolution on the different types of monocyte and macrophage populations that existed in that broader original category. One last type of plot I want to mention is called a pseudotime plot, which we can use to reconstruct cellular trajectories to infer developmental progression. All right, so here's the experimental setup for our study. So we performed a comparative analysis of respiratory, fungal, and viral infection using C57 black 6 mice infected with 500 arthoconidia of C. Posadasii, or BALB/C mice infected with 200 PFUs of influenza A. And then we monitored weight loss over time in order to select early, middle, and late time points for analysis, as we're interested in looking at how the immune response progresses over the course of disease. And so for the cocci-infected mice, we looked at animals at days 5, 9, and 14 post-infection, and for influenza, days 1, 3, and 6 post-infection. And so we harvested and pooled cells from lungs and blood of 3 different mice at each time point, and then subjected that to single-cell RNA sequencing. So we generated from this a total of 16 single-cell RNA sequencing data sets. And so obviously there's a lot of data and a lot of things that we could look at in these data sets. And actually we did publish a paper on doing a deep dive just on the cocci lung single-cell RNA sequencing data set. So if you're interested in checking that out, please do. But so for the purpose of this talk, I'm going to highlight 3 features of our data, including some myeloid cell differentiation in the lung, some unique macrophage populations that we identified in the cocci blood, as well as highlighting some neutrophil diversity and functional targeting. Okay, so here's a UMAP plot showing a merged immune cells from all the different data sets that we looked at. And I'm going to highlight just these 2 cell clusters, so the macrophage monocytes and the neutrophils, which I'm going to mostly talk about today. And that's because within both infection models, you see that these are both of these cell types are really dominate the lung immune landscape during infection. So we can look at how the relative abundance of cells changes over time. And so you see both in cocci and flu, the monocyte macrophage populations spike up early. And within cocci, we see a huge accumulation of neutrophils in the lungs, especially at that day 14 time point. And neutrophils also accumulate quite substantially in the lungs of the influenza-infected mice. Okay, so now I'm going to do We show a subclustering analysis of that monocyte macrophage compartment of cells. And so what you see here are the monocyte macrophage subclustered population separated out by pathogen type. So on the left, we have the subclustered cells from the cocci-infected animals. And then on the right is the influenza-infected animals. And that is paired with a pseudotime trajectory plot so we can look at how the cell populations differentiated over time. And so in both data sets, you see classical and non-classical monocytes, which are there highlighted in the dark green and light green. We also see interstitial macrophages, which are a population of lung-resident macrophages. And then within the cocci-infected lungs, you see the emergence of this large population highlighted in blue of macrophages that express high levels of this SPP1 gene. We also see a smaller population of macrophages that express high levels of CD5L. So in contrast that with the influenza-infected lungs, we don't see any macrophages develop outside of those interstitial macrophages, which are, again, lung resident. Instead, what we see are accumulation of a monocyte population that just is acquiring an inflammatory phenotype. And so by coupling the subclustering analysis with the pseudotime trajectory analysis, really the take-home message here is that classical monocytes in the lungs of the cocci-infected animals differentiate primarily into SPP1 high macrophages, whereas in the influenza-infected lungs, we see classical monocytes remaining as monocytes but acquiring a more inflammatory phenotype. Okay, so what are these cells doing in the lungs? So we did, well, we don't know, but just to do some functional enrichment analysis to try to gain a better understanding of how these cells are responding to infection. So the monocytes during influenza infection are acquiring this more antiviral phenotype, which is associated with type 1 interferon response, which is expected as an attempt to control viral replication. In the cocci lungs, the SPP1 macrophages and the CD5L macrophages are expressing a lot of genes associated with increased metabolism, as well as genes associated with fibrosis and tissue remodeling. So this suggests that these macrophages might be involved in sort of like lung damage that you see during cocci infection, as well as genes associated with hypoxia. So more than any other cell type that we looked at in our data, it seems like monocytes, their fate of monocyte differentiation is really shaped dramatically by the contrasting lung microenvironments that they see during viral versus fungal infection. All right, so now I'm going to talk about these cells in the blood. So this is a merged UMAP of all the blood cells that we sequenced in our different data sets, and here is how those cells fluctuate over time. So the most striking thing that you'll notice in this data is that both in the cocci and the flu-infected animals, you see by late-stage infection, there's this really dramatic expansion of neutrophils in the blood. But again, I want to highlight the monocyte macrophage cells here, because if you look particularly at this middle time point, we also see a pretty substantial expansion of a macrophage population within the blood of cocci-infected animals that you don't see in the influenza-infected animals. So again, when you subcluster that cell population, we see, again, classical and non-classical monocytes, as well as the SPP1 and CD5L macrophages. And when we look at the same UMAP, but color-coded by sample type, you see that the uninfected and the influenza-infected cells are both overlapping here, primarily in that, well, exclusively in the classical and non-classical monocyte compartment, whereas the cocci-infected sample is the CD5L and SPP1 positive macrophages are unique to the cocci blood. So these distinctive macrophage populations arising during cocci infection are detected both in blood and in the lung. Okay, so quickly now I'm just going to highlight again the neutrophils, because as I mentioned, they represent a very large component of the cells that we sequenced in the lung and the blood with both pathogens. So doing a subclustering of the neutrophils in the blood and the lung reveals diverse populations. And just in the interest of time, I'm not going to walk through all of the populations, but I want to highlight here that we noticed a population of neutrophils specifically in the lungs of both cocci and flu-infected animals that expressed very high levels of PD-L1. And so, given PD-L1's well-known role in immunosuppression in, many different contexts, we wanted to know if the PD-L1 expression here in the lungs of the cocci-infected animals on neutrophils was contributing to immunosuppression during lethal cocci infection. And so to test that, we again infected animals with 500 arthoconidia of C. Posadasii, and then treated the animals with 200 micrograms of either isotype control antibody or anti-PD-L1, starting at day 4 post-infection and then continuing every 2 to 3 days after infection, and survival was tracked through day 26. In a separate cohort of animals, we looked at PD-L1 expression on neutrophils at day 13 post-infection, and we see that indeed PD-L1 expression is decreased on neutrophils in the anti-PD-L1 treated group. And for survival, we noticed a slight but significant improvement in survival in the animals that were treated with anti-PD-L1. So the median time to death in the isotype control animals was 13 days, whereas in the anti-PD-L1 group, it was 15 days. And we had 20% survivors out to day 26 in the PD-L1 treatment group, whereas there were no survivors in the isotype control group. All right. So to summarize, by using single-cell RNA sequencing, we identified this distinctive macrophage subset both in the lung and the blood during cocci infection. And using PD-L1 blockade, we were able to modestly improve survival from lethal cocci infection. So for future directions, we're really interested in defining what the functional role of the SPP1 and the CD5L macrophages might be during cocci infection, as well as investigating mechanisms of their trafficking and differentiation, like how, are they emerging in the lung and then trafficking to the blood, or are they emerging separately in the lung versus the blood, and what implications that has for infection. We also want to explore the potential for using these macrophages as circulating biomarkers of severe disease, as well as further dissecting mechanisms of immune suppression and the potential for using immune checkpoint blockade as a therapeutic strategy. Right, okay, so just to acknowledge the team of people that did this work, and thank you all for your time and attention, and I'll be happy to take questions.
Thank you.
Hi, really exciting stuff to start trying to get down to the nitty-gritty details here. I'm curious on your single cell data. So you did day 5, which should be 1 day after spheroidal rupture, day 9, which I'm assuming you called your middle, and day 14. Histologically, the first cells that are coming in there are neutrophils. So can you explain the discrepancy in your data where you're saying neutrophils aren't coming in until day 14? And then you have a predominance of monocytes and macrophages at day 9, because histologically, that's not what you see at all.
So I can't explain that discrepancy. I will say that in the study that we did the single cell sequencing on, we saw, and then we've done other studies where we've looked at cell infiltration by flow cytometry. And when I've looked at in other animals by those flow cytometry studies, we do see neutrophil accumulation happening a bit earlier. So it could be that the animals that we used in that study, it was an anomaly that we didn't see neutrophils coming in earlier, because I have seen earlier neutrophil infiltration in other animals. But in terms of like broadly why our data would be inconsistent with previous studies, I'm not sure.
Yeah, I mean, it's really dramatic. If you look at histologically, as soon as that spheroidal cracks, it's surrounded
so I will also
by
say,
neutrophils.
Yeah, I will also say that the sequencing chemistry is actually not that great at capturing neutrophils. So the fact that we are capturing as many as we are at those later time points is actually quite striking to us, and we may even be underrepresenting the total number of neutrophils that are there.
Yeah. I think that could be a factor in this. Thanks.
Mm-hmm.
Hi, thanks. Did you try aligning the data set to a cocci reference genome, and did you detect any cocci transcripts?
So the probes that we used for this are mouse genome specific, so they wouldn't be capturing the cocci genome. That was something that actually we talked about and we would really have liked to be able to do, but in order to, so there is sequencing that you can do where the probes are captured via poly A tail, which we might capture cocci genome that way, but in order to get the samples out of our BSL-3 for sequencing, we have to be able to fix them. And so that type of sequencing technology is not compatible with the PFA fixing. So we have to do it this way where we use the mouse-specific probes. But you could put in some COX, if there were COX genes that you were of interest, you could throw in COX-specific probes.
Probably at that early time frame, you're not going to find it. You don't have enough COXI there unless you were to enrich for regions of inflammation that are going to be really. It's kind of amazing you're getting that immunology is local, not global. So,
I mean, consistently in any study I've done, though, you really get a huge amount of immune infiltration into the lung. And the
no, but I mean to get to your COXI read.
yeah.
To your COXI
Yeah,
reads,
also
at those
the-
time points, you've got very small numbers.
Yeah, well, also the kind of cell isolation protocol and stuff that we use is specific for mammalian cells. So we might not be, pelleting COXI with the same kind of efficiency.
Yeah, you have to.
You have to rethink the experimental design for doing that. Yeah. We're trying it with viral probes though, because that's a little easier because the virus is all mostly intracellular. So we are actually trying to do that, adding some viral specific probes to our sequencing reactions and seeing what we get. Okay, thank you.
Dina, I have a question too. It's great talk. Okay, we have identified the MDSC myeloid-derived suppressor cell, which also have the Ly6G positive marker on it.
I remember the talk from last year. It was quite good.
So and then we show that they are immunosuppressive for T cells. So I'm wondering whether you have looked into those T cells and whether they behave normally or they are suppressed.
Are you talking about in general or within
the
anti-PD-L1
14 mouse that you have?
Yeah, so within the data set that we have, the T cells are not very active at all. So that was a feature that we found in our COXI lung sequencing data where the T cells are not activated there. And actually you see sort of a decrease in you see a little bit of a boost of their activity at day 5, and then it really just decreases at the day 9 and day 14 time points. So yeah, very, very dismal T cell responses in this case.
Thank you. But I think I can pick up one of her neutrophil delay of infiltration into the lung, because our data also show that non-vaccinated mice, their neutrophil infiltration is delayed compared to the vaccinated mice. But they will suddenly increase in day 7 and day 14 to very, very high. So that's a phenomenon that we have observed in the lab as well.
Okay, good to know. Thank you.
Next up, we have Olivia Hepworth, single cell analysis and functional exploration of human airway epithelial responses to coccidioides posadasii infection.